Hello, and welcome back to another Science with Sully. Today, I will be going into what's called a stimulatory G protein. G proteins are proteins that have an alpha and a beta gamma subunit. Oh, that looks a lot like an alpha, so let's fix that. There we go. Gamma subunit. So let's see what a stimulatory G protein does in a cell. So we have our beautiful bilayer cell membrane. We'll note that here. And our cell membrane has these G proteins, and G proteins, as you probably know, are multi-layered, and they pass through the lipid bilayer a few times. And so we have 1, 2, 3 loops that are inside the cell. These are the loops that are going to communicate with everything downstream. So we also have our subunits. They will be here. And when inactivated, these subunits are associated with the protein. So how does everything get started? Well, you have what's called a ligand. The ligand that's usually, but not always, used that activates our stimulatory, and we'll call this a GS receptor. So our ligand for GS receptors is usually epinephrine or norepinephrine. So what's going to happen? Well, this ligand will come in contact with the G protein and things will start to change. First, our alpha subunit and beta subunit will dissociate. The alpha subunit has a GDP attached to it. So they're going to dissociate to where we now have our alpha subunit and our beta gamma subunit. Well, this alpha subunit interacts with another protein that changes its GDP to GTP. The alpha subunit is what's called a G protein. It is now active. So, what's going to happen when it's active? Well, the alpha subunit will go towards the cell membrane, and there's another protein that's waiting nearby called adenylyl cyclase. Adenylyl cyclase. And this protein does one very simple thing. It takes ATP and changes it to cyclic AMP, as we see here. ATP passes through and becomes cyclic AMP. So the alpha subunit is stimulatory of adenylyl cyclase, raising our cyclic AMP concentration in the cell. Well, the cyclic AMP concentration is going to do something else intracellularly. It is going to stimulate, so we'll write a plus here, another protein called protein kinase A. Protein kinase A. And Protein kinase A is a kinase, and kinases are enzymes that phosphorylate proteins. So it is going to increase protein phosphorylation. And protein phosphorylation could activate some proteins. It could be inactivating some proteins. Phosphorylation is different on proteins, but the big picture is this GS receptors increase protein phosphorylation. How do we keep this in check, though? There always needs to be a check and balances. Well, once proteins are phosphorylated, they need to become unphosphorylated eventually. So what happens? An enzyme will be added here, and it is called a phosphatase. And when the phosphatase is added to the pathway, we go back to normal. Well, we still have really high cyclic AMP. How do we fix that? Well, we've got another enzyme called phosphodiesterase. And that will break the diester bonds that are making cyclic AMP cyclic and convert cyclic AMP back to AMP. And then AMP, through other reactions, can go back to ADP. Now, where in the body could one of these GS receptors be found? Well, how about our beta-1 receptors in our heart? So, if you think about our ligand being epinephrine and the receptor being affected in the heart, when epinephrine hits one of these beta-1 receptors in the heart, it increases our heart rate. So this seems like a lot, but if you break it down and use, I'll be sure to mark, so when you try to do this on your own, this is where we start. You start with your ligand and then work your way down. I hope this video is helpful for you, and be sure to follow and subscribe to my channel for more videos that will be coming out.